Okay, everyone, we're going to go ahead and get started with our afternoon session. I hope everybody enjoyed lunch and had some time to look at the posters and stretch your legs a little bit, maybe warm up. So because this is a student-run event, we wanted to put an extra emphasis on student research. And so through the registration process, we encourage students to submit abstracts either for poster presentations or oral presentations. And we selected 3 students to present their research today for you. And these students will also be receiving a monetary award for participating in the event, and that has been provided by the Aggies Commit Grant, which is administered by the Texas A&M Office of Graduate and Professional Studies. So while everybody's getting settled, I'll introduce our first graduate speaker, Francisco Gomez. Francisco is originally from Honduras and graduated from the University of Florida with his bachelor's in agriculture operations management. He is currently a master's student in the plant breeding at Texas A&M, working with Dr. Bill Rooney. Francisco's work focuses on applying biomechanics to measure the biomechanical properties of sorghum stalks, with the goal of predicting lodging resistance and detecting QTL responsible for these complex traits. He was the recipient of the NSF Bridge to the Doctorate Fellowship and was also awarded the 2012 Young Botanist Award by the Botanical Society of America. After graduation, Francisco plans to pursue a position in academia with the goal of enhancing crops to maintain genetic diversity and global food security. Please join me in welcoming Francisco to the stage.
Right. Howdy. It's a pleasure to me to present my research today at the first Texas A&M Breeding Symposium. I'd like to thank my colleagues here for organizing this wonderful event. And what I'll be talking today about is applying biomechanics to predict lodging in sorghum. Now this is a picture of a cross-section of sorghum stem stained with fluoroglucanol. It stains the lignin molecule in the cross-section. Now the team is composed of a multidisciplinary team. Dr. William Rooney, he's a plant breeder. Dr. Anastasia Muliana, she's a mechanical engineer at the Mechanical Department of Mechanical Engineer at Texas A&M, and Dr. John Mullet, geneticist and biochemist. So there are 3 important points I want to discuss before I give this talk, and those 3 points are, physical laws and processes have routinely shaped the course of plant evolution throughout natural selection. So we can imagine an example of this is simple unicellular life forms have evolved to complex multicellular organisms, such as plants today. The second point is one of the greatest challenges the plant breeders have are the trade-off that results from trying to maximize the yield while simultaneously trying to reduce the probability of lodging. An example of this is the reduced height in cultivars during the Green Revolution, as you are familiar with that. And the third point is that biomechanics offers us a probe tool to quantify these biological materials and measure the biomaterials. They can allow us to help understand how plants and their materials evolved through natural selection and evolution of adapting to their environment, and we can use them to measure these biomaterials to predict lodging. So the crop I'm working with needs no introduction here at Texas. I'm working with sweet sorghum, and Dr. Jessup gave me a really good introduction for bioenergy. I think I can piggyback on his presentation on how important bioenergy is for the United States. And another characteristic of sweet sorghum is its high sugar and juice concentration in its stalk. That is what you see right here, the stalk of the sorghum. And so sorghum can grow 8 to 10 feet tall or taller, as you see in this picture. I'm actually 5'9", so you can calculate that. And sorghum has a great water use efficiency, a drought tolerance, and can yield up to 18 to 30 metric tons per hectare of dry biomass. So you can tell by the size brings some problems. So by increasing the size, we have increased the susceptibility for lodging. Now, Dr. Yu expressed he saw lodging somewhere. He saw lodging, root lodging. So that's one of the types of lodging we see in sweet sorghum. Sweet sorghum, root lodging usually occurs at the base of the plant when the plant uproots, usually after a storm event. And the second type of lodging is stem lodging, which is a mechanical failure at the stem where the plant breaks. Now my research is focused solely on stem lodging, particularly because it's sporadic in nature, which meaning when I began my research thesis, I did not see any stem lodging. So I grew impatient and I decided to we need another trait. We need to find a trait to be able to select for stem lodging, and a trait that will allow us to predict for stem lodging. And the way that I came up with was I needed to find the strength of a plant, and I did research on how do you measure the strength of a plant. The way you measure the strength of a plant is applying the laws of mechanics, so applied biomechanics to plants. Now this is a picture of a bridge, so in engineering, engineers to design a bridge, they figure out what materials we need to make the bridge of. So an example is steel, and steel has particular material properties, and from then they can decide how much, depending on the materials, they can decide how much stress will that bridge resist over time or can be applied to. And from then on, they can make the dimensions of the bridge. So we can take an engineering approach and take a very simplistic model, and we can think of the plant as a system. We can think of the stem as its structure. But in this case, in plants, the stem is a composite material composed of many other materials. As I mentioned to you before, it's lignin, cellulose, glucose, etc. So when we apply biomechanics to plants, what we're really interested in is the same thing the engineers are interested in, the material properties. I will exchange material properties with biomechanical properties in this presentation. So the biomechanical properties we're interested in is elastic modulus and strength. But at the same time, we want to look how those material properties behave under different geometries. So when I take stem lodging nodes, which I saw stem lodging in 2014, I started seeing a trend in where these plants were breaking, and these plants were usually breaking around internodes 3 and 6, so right in the middle of the plant. So I asked myself this question: how do we measure biomechanical properties in stem, in sorghum? What's the weakest section of the plant, and how does geometry affect the material properties of a plant? And with this in mind, we designed the first experiment where we applied a 3-point bend test, which is a way you can determine the mechanical properties of a plant. The objective was to determine the weakest section of the stem through characterization of geometrical and biomechanical properties of 6 sorghum genotypes. We put these genotypes because they varied in lodging characteristics as well. So we designed a randomized complete block design of 6 genotypes, 4 replications in 1 environment. We harvested 4 plants from the field. We measured their internode length, diameter, and we applied them to a 3-point bend test under 2 different loading configurations described by Vogel. So the first loading configuration is applied in force at the internode. So that's a 3-point bend test. So we're applying, this is a force gauge, this is a micrometer we're designed to measure the bending. We applied an internode and we applied the force, and this allows us to measure the after some calculations, we measure the biomechanical properties of that particular internode. The second loading is applying 2 internodes together, and we applied the force at the node. So that allows us to measure the biomechanical properties at the node. Data was collected, the data was cleaned, trait analysis was performed, and appropriate statistical models were run. We used LS means and took a comparisons test. So a few of the parameters I'd like to go over before I show my results, because they're going to be shown. Is the first parameter is slenderness ratio. Slenderness ratio is how slender something is. For example, we'll take an internode as an example. So the highest slenderness ratio an internode has, that means the formula for slenderness ratio is length over diameter. That means the ability that plant can bend more easily. So the second parameter is now a material properties is elastic modulus. Elastic modulus is the indication of material stiffness, so how stiff that material is, how stiff that stem is. The second mechanical property is strength, so the stress that will break the material. So we can say how strong that material is, which is what we wanted to figure out. And these are the formulas to determine elastic modulus and strength, which I will not go over. But I will say this is a very important point, that in order to calculate these formulas, we need to eliminate geometry. What that means is that these parameters are dimensionless. And this allows us to compare plants with different geometries. So this is very useful in this, because you might say, well, this has different geometry, it's going to be stronger. No, we removed that geometry to these equations, and they're not. And the fourth point is the product of elastic modulus times I, which is the second moment of an area, which is the cross-sectional area of the sorghum stem. And this tells us the ability to resist bending. Now we're taking the material property of stiffness, and we're multiplying times the geometry, and it gives us how the plant can resist to bending. Okay, so for my results, I'm sorry, this is a very busy slide, but the following graphs are going to be arranged this way. I have genotypes to my right. They're stacked one on top of another. There's 6 genotypes. And at the bottom, I have internodes. So this would be the first internode from the floor. So the bottom of the plant moving up. Now we will look at the geometry for internode length of a plant. We tend to see that plants tend to have a higher internode length around internodes 3 and 6. And we tend to see that plants have a slight uniform internode, but a slight decrease, but tend to be very uniform around the middle. And then for internode slenderness ratio, which is length divided by diameter, we tend to see that plants tend to be a lot more slender in the middle of around internodes 3 and 6, meaning those internodes can bend more easily. So, okay. Moving to the material properties for elastic modulus or Young's modulus, we tend to see a pattern around internodes 3 and 6, that we tend to see that plants have a higher Young's modulus around internodes 3 and 6. Now I put this plant to better picture that. You can imagine this plant did not have so that means this plant is stiffer at the middle. Now if this plant were not stiffer at the middle, what plants are doing, they're stiffening their material properties, because this plant needs to grow up to the top and needs to be erect. So that allows us to see this in plants and be able to see that plants make themselves do this material properties to stand up. And when we see for the parameter for flexural stiffness, we tend to see that plants have a higher flexural stiffness around internodes 3 and 6. Again, this makes sense because the plants are growing, competing for light, and they need to stand up. If that plant were not stiff and did not have a high flexural stiffness, you see this plant bending over. That's what it would do. Now we will look at the parameter for strength. We tend to see the opposite. So we tend to see a dip in strength. So we tend to see that plants are weaker around internodes 3 and 6. Now I mentioned to you before when we took lodging notes, I tended to see a pattern that plants usually have breakage around internodes 3 and 6. This is a picture I took out last year in 2014 when I was taking lodging notes. Plant breaking at internode 5. This is how they usually break, 1 or 2 inches above the node. I mentioned to you before that we had 2 different loading configurations. I've only showed you this graph for loading configuration number 2. We found out that breaking the biomechanical properties of the node for strength, it tells us that the nodes are stronger in the plant than the internodes. And this makes sense because we see plants usually break at the internodes than at the nodes. And this makes sense because plants over time have introduced those internodes not only to produce leaves, but as a reinforcement for the stem to grow. So with that knowledge, we know that the plants are weaker at the internodes, specifically internodes 3 and 6. And we asked ourselves a question, can we do this in a larger experiment? So we moved on forward and we applied this same procedure, but now only looking at internodes 3 and 6 because it requires less work. And we applied it to 15 genotypes varying in maturity. We designed a randomized complete block design, 4 replications in 3 environments, 1 in Weslaco, Texas, and 2 in College Station, Texas, planted 1 month after each other. So what we found in these 15 genotypes is that 1 genotype stood out of all the other genotypes, and this genotype was SOR 2014. this genotype had a tremendous Young's modulus. That means that this genotype is stiff, is very, very stiff. But now when we look at flexural stiffness, if you remember the formula for flexural stiffness is E times I, it has the lowest flexural stiffness, meaning that the geometry does not contribute much to that. So this plant can bend very easily. If you can imagine that plant in the field, it will move very easily like with the wind. So we'll not have any trouble moving like with the wind. Now when we look at internode slenderness ratio, it tells us that again that this plant has the highest slenderness ratio, this particular genotype, SORT 2014, and it can easily bend very easy. But at the same time, it has a highest internode strength. So this plant was the strongest among all those 15 genotypes. And we know for a fact that in our breeding program, this particular genotype, SORT 2014, is the most extremely lodging tolerant that we have in our program, and we're currently using it as a parent for our hybrids because it's extremely tolerant to lodging. So what this tells us is having a combination of high Young's modulus, being stiff, low flexural stiffness, high slenderness ratio, and high strength is a perfect combination for a lodging resistant plant. And with this, I conclude that applying a 3-point bend test, we were able to identify the weakest section of a sorghum stem corresponding to internodes 3 and 6. Plants tend to be stiffer at the nodes and stronger at the nodes than at the internodes. The experiment confirms that the observed point of stem breaking in the field of internodes 3 and 6 is due to their specific geometry and material properties. The application of a 3-point bend test appeared to have a potential to predict standability. So ongoing and future work is testing hypotheses, map QTLs for these traits, as we're currently working on with recombinant inbred lines, association between traits, and high-resolution image stem analysis. And with that, I'd like to thank my committee members, Dr. William Rooney, Dr. John Mullet, Dr. Anastasia Moliana, she's helped me a lot in the engineering part, and Dr. Carl Nicholas from Cornell University, who's an expert in plant biomechanics. He has contributed to a vast amount of knowledge to my project. And my graduate colleagues, the plant breeders who are sitting over there at that table, thank you for their support. Postdoc Dr. Leo Hoffman, and the student workers, which without them, I could not have done this project. Program technician Stephen Labar, who designed the 3-point bend test for me, and Derwood Collins. And with that, I'll take any questions.
We have about 5 minutes for questions, if anybody has any.
Take over. Guess not.
Oh, there's one in the front here.
How prevalent is stem lodging?
How prevalent? The numbers I can't get to you. I've been trying to get those numbers, but we've seen it last year was more prevalent than in 2013. So yes, it's prevalent in our fields. So what's useful, the question is, how prevalent is it? So it's good to understand how this, why those plants occur. So because when we breed for bioenergy sorghum, we'd like to know, so what do we need to, what are the trade-offs we can have while breeding, while designing a crop? We need to have an end goal. We need to imagine how we want this plant. So that's what this allows us to do.
So when you observe stem lodging, is it within the entire plot of that genotype?
Percentage, maybe some 70% of the genotype sometimes, yeah. And Weslaco particularly, I saw that, yeah. Ahora.
Francisco, in English, okay, but do you guys plan to test this model on segregating populations or on breeding populations to see if this is indeed effective for selecting?
We did this last year too.
Do you have any outcome of that already, or?
We have found some prevailing QTOs for that, so, but we're still adding more populations and more environments to that. So we have seen that. A similar study that I might mention is a study from partners from Monsanto and the University of New York University, I believe. So they did a similar study in corn. So if you read it in maybe 2 months ago in Crop Science and another one in the American Journal of Botany, similar results. They've done the 3-point bend test and found the working at the nodes is stronger than at the inner nodes. So it's a developing procedure. I think this has opened up a lot of doors to measure that type of lodging in both corn and sorghum. So.
Gracias.
We have time for one more question if anyone has any. And if anyone on the webinar wants to chime in, please type your questions now.
Okay, interesting work. There's a reason why we called Iowa stiff stock synthetic population, because
Yes.
the early maize breeding, it's stiff stock, right? You have to stand, carry the head. In your work, I'm curiously, because when we work with a biomass sorghum or sweet sorghum in the past, that is stem lodging, maybe because we were late in measuring the trait, or it is actually true that when you see the stalk breaks, you always see the pathogens like disease that somehow involved.
Stem borers as well, yes.
Yeah, I mean, I wasn't sure that in your case of this engineered testing, this particular variety, sweet sorghum, is also less disease susceptible?
No, we have the 15 genotypes that we put out have a diverse background in that we can see some diseases on them. We see stem borers on them. So pests attack them too. And I've been out to the field and being able to done enough, my advisor calls it forensic, basically doing an autopsy on the plant, because I've been out there when they break it. I discard disease, discard pests on them. So.
We have one question from a webinar attendee in regards to what is the biochemical property of stiffness underneath these genotypes?
The biomechanical properties of the stiffness underneath the genotypes. Oh, biochemical.
Biochemical, yes.
Oh, the biochemical. So we've done NIR, which, you took out the slide. Actually, that's the one, yeah. So we've done NIR composition. This was just a slide at the end, but we have a lignin. We try to figure out if lignin is correlated with strength, which is not. There's a lot of ways that strength, the biomechanical properties can associate with the biochemical properties of the stem or the composition of the stem. So that's what we're currently ongoing, trying to figure out how much lignin can contribute to cellulose. But we found out too that the arrangement of the particular molecules, for example, lignin, does affect the way that gives it the strength. And I've been told that in order to measure that, you have to do crystallography on the actual molecule, how that molecule is arranged. So that contributes to the mechanical properties.
Thank you, Francisco.
Sure, thanks.